Transcription and Translation by Ashley and Jessica. So let's start off with transcription. Transcription turns DNA into RNA so that it can leave the nucleus to make proteins for the cell. There are 4 DNA nucleotides. There are 4 RNA nucleotides too. The only thing that changes is thymine turns into uracil. The nucleus, where the transcription magic happens. What are the factors of transcription? DNA, RNA, DNA unwinding, promoter, RNA polymerase, cell's nucleus, and a cell's cytoplasm. The promoter is a sequence in the DNA strand where the transcription process begins. It has a sequence called the Tata box, T, A, T, A, 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 A.
The Tata box is where the DNA starts to unwind.
RNA polymerase attaches to the DNA, causing the DNA strand to unwind. As the polymerase moves, it reads and synthesizes a complementary messenger RNA. Except in that RNA, A pairs with uracil. A termination signal causes the polymerase to detach when the messenger RNA is formed. DNA then returns to its original double helix form. The 5' cap of the messenger RNA has 1 guanine leading it. The 3' poly A tail has 250 adenines tagging along. Extra misleading information in the messenger RNA gets cut out in the splicing process and gets put together. The good stuff is called exons, and the junk that is then reused in the nucleus is called introns.
The DNA template strand is the strand in which the RNA polymerase is reading it off and making the mRNA.
Uracil is added to the RNA by bind. It is paired with adenine on the template DNA strand. A phosphodiesterase bond is formed. The RNA chain is then elongated by 1 nucleotide. DNA plus RNA polymerase equals DNA-RNA hybrid complex.
A mutation is when one of the bases is changed in the incorrect way. A mutation can have multiple results depending on how many times it changes and what changes too.
So now let's move on to translation. Translation takes the messenger RNA we just created in transcription and uses it to create amino acids in our cells. Translation happens in the ribosomes floating around in the cytoplasm in the cell.
tRNA transports amino acids to the ribosome during translation. This is the codon. This is the anticodon. The anticodon finds the match for the codon and based off of that, they make a protein up here, aka amino acids. The start codon is the first codon that is translated.
So in our ribosome, we have 3 sites. Our transfer RNA will come into the first site, and the easy way to remember it is the amino acid site. It is called the A site. There are 3 anticodons will connect to these 3 codons. As soon as it comes in, it will move on to the P site. Also, an easy way to remember is the polypeptide site, where the proteins will keep building up. Then it will move on to the E site. And easy to remember is the empty site, where they will leave and their amino acid will join onto the polypeptide chain. They will keep coming in and keep moving on until the chain gets bigger and bigger. Elongation is the stepwise addition of amino acids to the growing protein chain. The order of amino acids is specified by the sequence of codons in the messenger RNA, the genetic code, protein factors. Eukaryotic elongation factors are also necessary to speed up the elongation cycle.
The mutation in translation results in a protein changing its function because when the mRNA is making the proteins, there will be a change in base, which means there's a change in protein, which will have a different protein made.
Thanks for watching.